Welcome back, folks, to our second class where we're discussing enzyme inhibition, but today we'll also talk about allosteric control. Here are today's learning outcomes. So first, we're going to learn about what allosteric control is. Then we're going to learn about two different case studies. The first with an enzyme called ATCase. We'll understand why a molecule called ATP activates this enzyme and a molecule called CTP inhibits the enzyme. And we'll learn a little bit about ATCase's quaternary structure. The second example we'll learn today is glycogen phosphorylase. So we'll understand about allosteric modulation of glycogen phosphorylase and also how some various small molecules inhibit or activate that enzyme. And again, we'll connect changes to this enzyme to its structure as well. Today's Meet a Scientist moment is Asifa Akhtar. She is a senior group leader at the Max Planck Research Institute. She's of Pakistani descent. She studies lysine acetyltransferases that act to acetylate histones. And so this is a particularly important example of how gene regulation happens, right? And that is not a focus of this class. That would more be a focus of Biochem 2, and in particular, probably a genetics course or a cell and molecular course. But since we're here to talk about regulation, I thought it was a great connection to research being done today. So let's jump into it. What is allosteric control? It is modulation of an enzyme's activity at the active site by binding of a molecule somewhere else. So, binding not in the active site. And this can be by a small organic molecule or a protein or something else. Today we're going to talk about both activation of enzymes and inhibition of enzymes. So last time we were together, we talked about how inhibitors are going to reduce Vmax. Well, today we'll be talking about activators, and in particular, we're going to see an example of how an activator is going to decrease the Km. When we draw activators and inhibitors into this type of reaction scheme, we draw the activator with an arrow pointing directly towards the enzyme, or an inhibitor with a blunt-ended arrow pointing towards the enzyme. And this shows us either activation or inhibition. Up next, we'll talk about the first enzyme example, ATCase.